Thank you everyone for returning from the break. And of course, thank you, Nathan, for agreeing to present here. So it's my pleasure to introduce Nathan West. Nathan is currently a principal scientist in the Department of Cancer Immunology at Genentech, where he studies the roles of both immunosuppressive and immunostimulatory cytokines in the context of various diseases. In the case of today's talk, IL-6 in the context of resistance to checkpoint blockade and therapy. Nathan has always been a cytokine researcher. He completed his PhD research at the University of British Columbia Cancer Agency in Victoria, where he studied the impact of inflammatory cytokines on breast cancer production or progression, and of course, the relationship between anti-tumor immune responses and chemotherapy. In 2012, Nathan moved to the University of Oxford for his postdoctoral training, where he continued to focus on cytokine biology, but pivoted away from cancer and really dove into inflammatory bowel disease and was characterizing novel genetic correlates and cytokine drivers of inflammatory bowel disease and also colorectal cancer. He joined Genentech in 2017, and Nathan, we're looking forward to hearing your talk.
Great, thanks for the intro, and thanks to the organizers for inviting me to give this talk. Yeah, it's a real pleasure to be here. So as Jonathan kind of alluded to, that this talk is going to be a slight change of pace compared to the previous two, because I'm actually not going to talk about engineering anything at all. So I hope that's not a huge disappointment. This is going to be more about looking at cytokines from kind of the opposite perspective. So in this case, thinking about drivers of resistance to existing immunotherapies and how we can overcome them. And in this case, obviously, it'll be about IL-6. So I just want to start by acknowledging a lot of people who've been involved in this, particularly folks highlighted in red. So Li-Fen Wang in my lab, who's a super hardworking and talented senior scientist. She did a lot of the bench work associated with this study. And also particularly our colleagues in our biomarker development group who've played big roles in looking at clinical associations between IL-6 and clinical outcomes to checkpoint blockade. So, the key kind of challenge I'm going to talk about in this presentation is the issue of trying to overcome resistance to checkpoint blockade therapy. So as you guys all probably are very familiar with, checkpoint blockade is by far the most standard form of immunotherapy for cancer currently. But it's not nearly as effective as we would like it to be. Certainly there are some indications that are much more responsive than others, but even within responsive indications, it's usually only a minority of patients that have significant benefit from checkpoint blockade. And I think that's related to the fact that driving an anti-tumor immune response is a very complex process, right? And with checkpoint blockade therapies, we're hitting some aspects of that. In this case, we're liberating signaling through key drivers of T cell activation, like T cell receptor signaling, CD8 signaling, by targeting negative regulators like PD-1 or PD-L1 or CTLA-4. But this is just kind of the kind of early steps involved in T cell activation. And while very important, there are plenty of other factors that can influence a T cell response. And, the subject of today is a great example. What's not represented here is the so-called signal 3 associated with T cell functional differentiation and maturation. And in many cases, that signal 3 takes the form of cytokine signaling. So, a key challenge for us is out of all these various options represented in this wheel of resistance, can we identify sort of actionable drivers of therapeutic resistance that we can actually exploit in the clinic? So at Genentech, and similar to a lot of other companies who have access to big clinical data sets, we exploit the so-called bench to bedside kind of learning loop, where with growing experience in clinical trials, we now have access to a lot of clinical data from patients treated with various agents, including in Genentech's case, anti-PD-L1 or atezolizumab. And what we can start to do as we accumulate this data is we can, leverage various kind of omics readouts associated with these clinical trials, including patient genomics, tumor transcriptomics and circulating biomarkers and PBMCs and so forth, to try to identify factors that are positively or negatively associated with response to therapy. And as I've already mentioned, our goal is to identify resistance drivers that we can actually target in the clinic. So I'll just cut to the chase and get into sort of some of the early kind of biomarker data we generated that brought IL-6 to our attention here. So in this case, we were evaluating a clinical trial of previously untreated metastatic renal cell carcinoma called Emotion 150. And a couple of key points about this trial is that, as I said, these patients were previously untreated. So we're looking at sort of, the baseline biology. It hasn't been influenced by therapy yet. This was a randomized study in which patients were treated either with atezolizumab monotherapy, atezo plus bevacizumab, so anti-VEGF, in combination, or sunitinib as just a sort of pan, or not a pan, but a multi-tyrosine kinase inhibitor kind of control arm. So within the study, we can actually look at factors that are maybe associated specifically with atezolizumab success versus some of these other therapies. And so the first approach we took was just to take tumor RNA-seq data where we compared patients who either had disease progression or had some evidence of disease stabilization, and just looked at differential gene expression. And one of the things that was immediately clear is that a lot of factors associated with tissue remodeling and kind of innate inflammation were associated with disease progression, including IL-6 and also its family member LIF. As well as one of the key factors downstream of IL-6 and STAT3 signaling called SOX3. And on the other side of the spectrum, actually, we have a lot of things associated with antigen presentation and interferon response associating with favorable clinical outcome, perhaps not surprisingly. When we look at the association between IL-6 and overall survival rather than just objective response, we see that this pans out the way we would expect. So within the atezolizumab monotherapy treated patients who have high IL-6 expression are clearly doing worse relative to those who have low IL-6 expression. And what's interesting is that this relationship is not nearly as clear in these additional arms. So there's a bit of a trend when you have bevacizumab added on. It's not statistically significant, and then there's really no relationship at all in the sunitinib treated patients. So this appears to be predictive of atezolizumab outcome rather than just prognostic. And if we look at IL-6 protein, we can't really do that within tumor tissue in these studies, but what we can do is look for IL-6 in circulation, so in the plasma from these patients. And something that was immediately apparent, and this is consistent with I think extensive literature over the years, is that cancer patients, especially with advanced stage disease, have very high circulating IL-6 compared to healthy controls. And we're seeing that here just based on trials performed in-house on 3 different types of cancer. So metastatic triple-negative breast cancer, metastatic renal cell cancer, and metastatic urothelial cancer, so bladder cancer. And in all 3 of these cancer types, if we look at patients who have high plasma IL-6, in this case defined by greater than 10 picograms per ml, we find that these patients are doing poor with atezolizumab monotherapy, irrespective of the cancer indication. So this seems to be a very generalizable phenotype. And what's interesting is we see very similar results if we just look at serum CRP, which is a very classic systemic inflammation marker, and importantly is very much IL-6 induced. So it's nice to see that as kind of a confirmatory correlate. So just in case anybody's not super familiar with IL-6, I wanted to touch on the signaling and kind of what's known about it already as a clinical target. So IL-6 can signal through 3 distinct mechanisms. The kind of best recognized one is the conventional approach where IL-6 is binding to cell surface receptors. In this case, it's a complex of the IL-6 receptor, alpha chain, and then GP130 as the actual kind of signal transducing receptor. What's notable, though, and important to think about whenever you're studying IL-6 is that there are these other mechanisms, including trans-signaling and trans-presentation. And these are both based on the ability of IL-6 either in solution or bound to another cell to bind to IL-6 receptor and be able to bind to GP130 either as a soluble complex or, like I said, when it's presented by another cell here. And so what this means is that you can actually get IL-6 signaling in cells that don't actually express their own IL-6 receptor. And so if you dig in the literature, you'll see a lot of reference to this process, and it's thought that actually this trans-signaling mechanism is a big driver of inflammatory disease. So downstream of receptor engagement, you primarily have activation of STAT3, although you can actually activate some other pathways too, such as the MAP kinase cascade and the APTAZ signaling. But as I'll show you in a bit, the STAT3 pathway is kind of the preeminent pathway for IL-6. And just to cover what IL-6 is currently used for, so I think probably the most widely used drug targeting the IL-6 pathway currently is tocilizumab or Actemra. This is a Roche Genentech Chugai drug. It's approved for rheumatoid arthritis, juvenile idiopathic arthritis, Castleman's disease, giant cell arteritis, and also more recently CRS for CAR T therapies. There are a couple of others out there as well, including sirilimab and siltuximab, both of which are kind of targeting similar sorts of conditions. And as you guys have probably seen in a few papers, there's a lot of attention on using anti-IL-6 agents to deal with some of the complications of severe COVID-19 infection. So how could IL-6 potentially impact anti-tumor immunity? So it's basically the definition of a pleiotropic cytokine. So there are many possible ways that it could do that. IL-6 is made by a lot of things, including myeloid cells, fibroblasts, and cancer cells. And it can act on many cell types, in part because of the ability to act through this trans-signaling mechanism that I just described. So in cancer cells, it's thought to promote proliferation and apoptosis resistance, various other things like EMT and stemness. In the hematopoietic compartment, there are many different functions ascribed to IL-6. Generally speaking, it's thought to drive kind of M2 or sort of immunosuppressive myeloid phenotypes. It's also well known to promote Th17, CD4 T cell polarization. And in contrast, it's thought to sort of inhibit dendritic cell maturation, IL-12 production, and thereby kind of inhibit Th1 polarization. It can also act through mesenchymal cells as well, and is implicated in angiogenesis, fibrosis, and potentially is involved in the MOA for TGF-beta. So it's involved in a lot of different things. We don't quite know if one mechanism is more dominant than the other for affecting anti-tumor immunity. And so we were interested in trying to understand this a bit better. So the first thing that we did was to leverage our mouse models. So in this case, we're using a system that Jeff actually described in his talk already, the EMT6 model. We like this for combination therapy questions because it's a partially responsive model to PD-L1 blockade. So blocking PD-L1 results in some tumor control, but it's not particularly strong, which means you can move the needle with the right combination. And we've looked at other models as well and seen similar results, including MC38, CT26. And the key take home here is that when we block IL-6 signaling using an antibody against IL-6 receptor, so similar to Actemra or tocilizumab, relative to anti-PD-L1 monotherapy, we can pretty dramatically improve animal survival by reducing tumor progression. Interestingly, IL-6 receptor blockade by itself really has little to no effect. So this is a clear kind of synergy rather than an additive effect when we look at it in combination with anti-PD-L1. So this is just progression-free survival. And then along the bottom here, we can see, individual tumor growth from a representative study here. Again, IL-6 receptor blockade by itself doesn't do a whole lot. Anti-PD-L1 delays tumor growth, and for a minority of mice, it can actually regress the tumors. But when we have combination therapy on board, we can substantially increase the frequency of complete tumor progression or partial tumor regression. And so, of course, we were interested in understanding what is actually happening in the tumor tissue associated with this tumor control. And so, we've done a lot of phenotyping of the immune compartment in mice treated with combination therapy. And in all the models we've evaluated, the one thing that sticks out that is consistent across the board is an increased CD8 T cell response. So IL-6 has not historically been thought to be particularly critical for CD8 T cell biology. And so it was striking to us that CD8 T cells were kind of the key pharmacodynamic readout for IL-6 blockade in combination with anti-PD-L1. So you can see here, this is just frequencies of different leukocyte subsets within total CD45 positive tumor infiltrating lymphocytes, or leukocytes, I should say. And although there's a slight reduction in macrophages in this particular system, the main thing that we see is a clear expansion of the CD8 T cell compartment. We see little or no changes in other myeloid populations or CD4 T cells. And what's cool is that we don't just see an increase in CD8 T cells, but their function appears to be substantially affected as well. So here we're looking at CD8 T cells after restimulation ex vivo and looking for effector function, in this case, expression of TNF, interferon gamma, and within TNF and gamma double positive cells, expression of granzyme B. And what we see pretty clearly is that in combination therapy treated mice, we have a clear increase in what we would call polyfunctional CD8 T cells. So cells that are co-expressing multiple effector markers like TNF, interferon gamma, and granzyme B. And this is quantified here. So with single agent therapies, the frequency of CD8 T cells with this polyfunctional phenotype is slightly boosted. It's statistically significant. It's not particularly profound, but then it's clearly boosted in the combination setting. And when we factor in cell expansion as well, and look at the total number of polyfunctional T cells within the tumor bed, we find that there's a, approaching a tenfold increase in those polyfunctional T cells in the combination therapy setting. So it's a pretty potent effect. This made us, ask whether we can see any evidence of altered CD8 T cell biology in cancer patients. And again, that's hard for us to do in the tumor tissue because we're kind of restricted to things like immunohistochemistry. So in this case, in order to profile T cells more deeply, what we did was evaluate circulating CD8 T cells from peripheral blood in cancer patients that were pre-selected for being either high or low for plasma IL-6. And then we did single cell RNA sequencing to profile their phenotypes. So in this case, we looked at two different kind of data sets, blood from renal cell cancer patients or blood from bladder cancer patients. And we kind of treated them as separate analyses, at least initially. Like I said, we did single cell RNA sequencing. We pulled out the CD8 T cells, and then we looked at genes that were associated either with low IL-6 or high IL-6. And what we found that was actually consistent between the two data sets was that CD8 T cells from patients with low IL-6 had clear enrichment of genes related to antigen presentation, interferon signaling, other types of cytokine signaling, and interestingly also sort of metabolic differences, particularly enrichment of OXPHOS and TCA cycle related genes. Whereas in the case of patients with high IL-6, their CD8 T cells didn't show enrichment of a lot of things, but what they did show enrichment of was stuff related to glucose metabolism and translation. And this is actually mainly just driven by ribosomal genes, which we interpret to mean a more kind of quiescent cell phenotype. And what's interesting is if you just drill deeper and try to do subclustering on these CD8 T cells, what's interesting is that we find evidence of polyfunctional effector T cells that are pretty much exclusively found in patients with low plasma IL-6. So this is, broadly speaking, kind of consistent with what we're observing in vivo in mice. So to look at this more directly, we've done a lot of bench work, both with human and mouse CD8 T cells to try to understand what IL-6 is doing. So in this case, this is OT1 CD8 T cells activated with peptide in the presence or absence of IL-6. And what we find is that their effector function is dramatically perturbed when IL-6 is on board. So they express a lot less TNF, a lot less interferon gamma, and reduced granzyme B. And consistent with this, if we try to use these T cells in an in vitro killing assay where we have peptide pulse target cells, when these T cells are activated in the presence of IL-6, as shown in this red line, they're substantially reduced in their or attenuated in their ability to kill target cells. So we've done some deeper profiling, including RNA sequencing of T cells activated with or without IL-6. In this case, this is using a kind of a mixed cell assay in which we take bulk splenocytes in order to provide antigen presenting cells, but also to provide an endogenous source of IL-6 and activate them with peptide, and then basically sort the CD8 T cells out afterwards and do RNA sequencing. And what we found is very similar to what I just showed you in the previous slide, but in a more kind of unbiased scale. So in the absence of IL-6 signaling, in which we block all IL-6 receptor signaling, the CD8 T cells really kind of skew towards a heavily cytotoxic phenotype with lots of cytokine expression, granzyme expression, and other cytotoxic factors. Whereas in the presence of IL-6, what's interesting is that they preferentially retain features associated with naive or central memory cells, including CCR7, IL-7 receptor, L-selectin, and TCF7. And we can kind of confirm this with flow cytometry. So if we stop looking at cytokines and look for other factors like transcription factors that drive effector function, including EOMAs and TBET, versus markers of more naive-like cells like CD62L, we find that in the presence of IL-6, the T cells really do kind of retain this naive-like state where they express high CD62L, but low amounts of these effector-associated transcription factors. And finally, if we take all of these genes and map them onto previously published kind of modules of genes associated with different stages of T cell differentiation, we find that when we get rid of IL-6, the majority of differentially expressed genes are associated with early stages of effector differentiation. Whereas in the presence of IL-6, the differentially expressed genes in kind of green and blue and teal are all associated with transcriptional patterns related to naive or memory-like cells. So we think that IL-6 is actually basically preventing the full transition of T cells into an effector state, which is likely limiting their potency for anti-tumor function. We were interested also in what might be driving this, and the first thing we did was to confirm that STAT3 was involved. And indeed, if we take T cells that are deficient in STAT3, we don't see this inhibitory effect of IL-6 any longer. We also did some RNA sequencing to try to identify early induced factors driven by IL-6 that might be responsible for this effect. So this is just T cells activated in the presence of IL-6 for 4 hours. And what we found that was interesting was a transcription factor called BATF that was quite profoundly induced by IL-6. And BATF has actually been shown by Nick Haning's lab and others to regulate effector differentiation by promoting cell survival, but at the expense of expression of effector molecules like interferon gamma. And so we were interested in knowing if BATF is involved in our system. And sure enough, we found that BATF is induced at the protein level by IL-6 and CD8 T cells in a STAT3 dependent manner. And if we use CRISPR-Cas9 to knock out BATF from primary CD8 T cells, we find that the effect of IL-6 on sort of preventing this conversion to an effector phenotype is largely reversed. So we think that at least one molecular mechanism for this is STAT3 dependent BATF expression, and that BATF then basically affects the chromatin reorganization in a way that prevents effector differentiation in these T cells. And I'll just finish up this by pointing out that we can actually show in vivo that this effect of IL-6 on CD8 T cell function is a cell intrinsic one. So obviously, because IL-6 is so pleiotropic, it could be acting through many cell types in vivo to drive the effects that we see in our tumor models. So what we wanted to know is if any of this effect is actually cell intrinsic in the CD8 T cell compartment. So what we did was generate mice that have IL-6 receptor deficiency specifically in the CD8 T cells. And this is using the E8I CD8 Cree system, where Cree is active only in mature CD8 T cells. And what you can see here is if you compare CD8 versus CD4 T cells from lymph nodes in this model, you find that IL-6 receptor is really exquisitely lost just in the CD8 T cells and is totally normal in other cell compartments. And when we implant tumors into these mice and treat them with anti-PD-L1, we find that the CD8 T cells that are deficient in IL-6 receptor respond better and start to regress these tumors. And you can see their phenotype up here in the flow cytometry plot. Again, in the absence of IL-6 receptor expression, these CD8 T cells respond to anti-PD-L1 by inducing expression of things like TNF and interferon gamma more aggressively than in their wild type counterparts. And at a total cell number level, we see a pretty clear difference in terms of improved CD8 T cell response in these IL-6 receptor deficient mice, demonstrating pretty clearly that although IL-6 might be acting through other cell types to some extent, this cell intrinsic signaling mechanism through CD8 T cells is clearly playing a role. And this is the last data slide I'll show. We also did RNA sequencing of these IL-6 receptor deficient CD8 T cells, and similar to what we see in vitro, we see pretty broad impacts on pathways like cytokine response, OXPHOS and TCA cycle, cell division, and various DNA and mRNA processing pathways that are all upregulated in the IL-6 receptor deficient T cells relative to the wild type cells, suggesting that these are more active, effector differentiated cells. So just to summarize, we see IL-6 clearly associated with lack of response to immunotherapy in various tumor types. We also see that high plasma IL-6 is related to CD8 T cell effector function in cancer patients, at least in peripheral blood CD8 T cells. This is actually very consistent with data that was published over the last few years from COVID-19 patients, where again, high circulating IL-6 is associated with exhausted looking CD8 T cells. And I should also mention that it's also been published recently that IL-6 is prognostically related to anti-CTLA-4 and anti-PD-1 therapy in melanoma patients. So our clinical data is consistent with the experience of others as well. So what we think is happening is that in the context of anti-PD-L1 therapy, T cells that are seeing antigen normally in the presence of IL-6 are activating and proliferating, but their acquisition of effector function is limited by this effective IL-6 on activating STAT3 and potentially BAD-F to block this full effector transition. When we get rid of IL-6, what we see is that these, at least in preclinical systems, is that these tumor infiltrating T cells acquire a much more potent effector phenotype and are more capable of driving tumor regression. And I probably went slightly longer than I should have, so apologies for that, but I'll stop there and take any questions. Thank you.
Thanks, Nathan. That was great. I will kick the Q&A off. I think we have time for a couple questions. If anyone wants to populate questions in the Q&A, feel free to do so. I'll kick it off by just saying, so what's the clinical landscape? I mean, blocking IL-6 has been around for a long time, as you pointed out. Is there any clinical landscape associated with looking at blocking IL-6 in the context of immune checkpoint blockade?
Yeah, I mean, it's a good question. I think so. Genentech for sure is looking into this, right? It's an easy question for Genentech to ask because we can just use our own IL-6 receptor antibody, of course, like Actemra, to ask these questions. But it's not just us. There are others as well. I think siltuximab, at least according to clinicaltrials.gov, is being used in combination with things like anti-PD-1 for tough indications like pancreatic cancer, for example. There are a number of different company and investigator-led studies that are starting to look at IL-6 blockade in the context of checkpoint inhibition. So people are definitely into it. I think it'll be interesting to see what happens. But I think the other thing too is that a lot of these IL-6 drugs are getting a bit long in the tooth, and so there's going to be an effort probably to try to revitalize and improve upon the existing anti-IL-6 agents. In order to sort of keep this thing rolling. Yeah.
One more quick question. Have you looked at timing of IL-6 blockade at all?
In the context of
PD-1 therapy, like, is there an effective
right.
timing?
So we think it has to happen concurrently. Based on, particularly from our in vitro data, we find that the critical events driven by IL-6 are basically concurrent with TCR stimulation. So if you activate a T cell and then wait several days and give them IL-6, the T cell actually doesn't really care. So basically what we're affecting is T cells at the point of activation, which is something that anti-PD-L1 promotes, right? So a key function of anti-PD-L1 is not just to restore function of already activated T cells, but to promote the optimal activation of, non-activated T cells, right, that are getting stimulated for the first time. We think that's actually the axis that IL-6 is affecting. So, so if you were to give a bunch of anti-PD-L1 and then cycle patients onto an anti-IL-6, I don't think that would work very well.
Yeah. Very cool. Well, I think in the interest of time, we did have one question, Nathan, just show up in the Q&A. So if you want to take care of that one, but I think in the interest of time,
Sounds
we'll
good.
move on. Thank you again, Nathan, for that great talk.
Thanks, guys.
And